Chapter 8. The Cell, the Unit of Life. When you look around, you see both living and non living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have which a living thing have, has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms. Like us, composed of many cells are called multicellular organisms. What is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw the described a living cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Sheldon, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissue of the plant. At about the same time, Theodore Sanborn, 1839, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, the, that the presence of cell wall is a unique character of plant cells. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Sheldon and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells. Omnin cellula is cellula. He modified the hypothesis of Sheldon and Schwann to give the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and products of cells. All cells arise from pre-existing cells.
An overview of cell. You have earlier observed cells in an onion peel and on human cheek, cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of human cheek have an outer membrane as in delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosome which in turn contain the genetic material DNA. Cells that have membrane bound nuclei are called eukaryotic, whereas cells that lack a membrane bound nuclei are prokaryotic. In both prokaryotic and eukaryotic cells, A semi-fluid matrix called cytoplasm occupied the volume of the cell. The cytoplasm in the main area of cellular activities in both the plant and animal cells. Various chemical reactions occurs in it to keep the cell in the living state. Beside the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like the endoplasmic reticulum, the Golgi complex, lysosome, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane bound organelle found in all cells, both eukaryotic as well as prokaryote. Within the cell, ribosomes are found not only in cytoplasm but also within the two organelles, chloroplast in plants and mitochondria and rough ER. Animal cells contain another non-membranous bound organelles called centrosome, which help in cell division. Cells differ greatly in size, shape, and activities. Figure 8.1. For example, mycoplasma, mycoplasmas, the smallest cells, are only zero point 0.3 nanometer in length, while bacteria could be 3 to 5 nanometer. The largest isolated single cell in the eggs of ostrich among multicellular organisms. Human red blood cells are about 7 nanometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, phylogenal, columnar, cuboidal, thread-like, or even irregular. The shape of the cell may vary with the function of they perform. Prokaryotic cells. The prokaryotic cells are represented by bacterial, blue-green algae, mycoplasma, and PPLO, pneumonia-like organism. They are generally smaller and multiply more rapidly than the eukaryotic cells. Figure 8.2. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, ochus, spherical, vibrio, comma-shaped, and spirillum, spiral. The organization of the prokaryotic cells is fundamentally similar even though prokaryotes exhibit a wide variety of shape and functions. All prokaryotes have a cell wall surround the cell membrane, except in mycoplasma, the fluid matrix filling the cell in the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have small circular DNA outside the genome DNA. The smaller DNA are called plasmids. The plasmid DNA confirms certain unique phenotype characters to such bacteria. On such characters is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryote cells except for ribosomes. Prokaryotes have something unique in the form of inclusion, a specialized differentiated form of cell membrane called mesosome in the characteristics of prokaryotes. They are essentially enfolding of cell membrane, cell envelopes and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of difference in the cell envelope and the manner in which they respond to the staining process developed by gram via those that take up the gram stain are gram positive and the other that do not are called gram negative bacteria. Glycocalyx differ in composition and thickness among different bacteria. It could be a loose sheet called the slim layer in some, while in other it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provide a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membrane structure is the mesosome, which is formed by the extensions of plasma membrane into the cell. The extensions are in form of vesicle, tubules and lamellae. They help in the cell wall formation, DNA replications and distribution to daughter cells. They also help in respiration secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into a cytoplasm called chromatophores which contain pigments. Bacterial cells may be motile or non-motile. In motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filaments, hook, and basal body. The filament is the longest portion and extension from the cell surface to the outside. Basidius, flagella, pili, and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made of a special protein. The fimbriae are smaller bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rock in stream and also the most tissues. Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits 50S and 30S unit, which when present together form 70S prokaryotic ribosomes. Ribosomes are the sites of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysomes. The ribosomes of polysomes translate the mRNA into protein. Inclusion bodies. Reserve materials in prokaryote cells are stored in the cytoplasms in the forms of inclusion bodies. These are not bound by any membrane systems and live free in the cytoplasm. Example: phosphate granules, synophysian granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Eukaryotic cells. The eukaryotic include all the protein the eukaryotes include all the protist plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organism nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell walls, plastids, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have controls which are absent in almost all plant cells. Animal cells have centrioles which are absent in almost all plant cells. Let us now look at individual cell organelles to understand their structure and functions. Cell membrane. The detailed structures of the membrane was studied only after the advent of the electron microscope in the 1915s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to deduce the possible structures of plasma membrane. These studies show that the cell membrane is mainly composed of lipid and protein. The major lipid are phospholipids that are arranged in bilayer. Also, the lipids are arranged within the membrane with the polar head toward the outer side and the hydrophobic tails toward the inner side. This ensures that the nonpolar tail of saturated hydrocarbons is protected from the aqueous environment. Figure 8.4. In addition to phospholipid membrane also contain chlorostol. Later, biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% lipid. Depending on the each of extractions membrane protein can be classified as integral and peripheral. Peripheral proteins lie on the surface of membrane while the integral proteins are partially or totally buried in the membrane. An approved model of the structures of cell membrane was proposed by Singer and Nicholson 1972, widely accepted as fluid mosaic model. Figure 8.4. According to this, the quasish fluid nature of lipid enable lateral movements of protein within the overall bilayer. The ability to move within the membrane is measured as the fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formations of intracellular junctions, secretions, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some molecule present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Natural solids may move across the membrane by the process of simplification along the concentration gradient, that is from higher concentration to lower. Water may also move across this membrane from higher to lower concentration. Movements of water by diffusion is called osmosis. As the polar molecules cannot pass through the nonpolar lipid bilayer, they require carrier protein of the membrane to facilitate their transport across the membrane. A few ion of molecules are transported across the membrane against their concentration gradient, that is from lower to higher concentration. Such a transport is an energy dependent process in which ATP is utilized and is called active transport. Example, sodium and potassium pump cell wall.
As you may recall, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell walls not only give shape to the cell and protect the cell from mechanical damage and infection, it also helps in cell-to-cell interactions and provide barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactose, mannan, and minerals like calcium carbonate, while in other plants, it consists of cellulose, hemicellulose, pectin, and protein. The cell wall of young plant cells, the primary wall, is capable of grow while gradually diminished as the cell mature, and the secondary wall is formed on the inner toward membrane side of the cell. The middle lamina is a layer mainly of calcium pectate which hold or glue different neighboring cells together. The cell wall and middle lamellae may be tra- traversed by plasmodesmata which connect the cytoplasms of neighboring cells endomembranous system. While each of the membranous organelles is distinct in terms of its structures and function, many of these are considered together as an endomembranous system because their function are coordinated. The endomembranous systems include endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. Since the functions of the mitochondria, chloroplast, and the peroxisomes are not coordinated with the above components, these are not considered as a part of endomembranous system. The endoplasmic reticulum. Electron microscope studies of erythrocyte eukaryotic cells revealed the presence of network or reticulum of teeny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum. Figure 8.5. Hence, ER divides the intracellular space into two distinct components, that is luminal inside ER and extraluminal cytoplasm compartment. The ER often show ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called a rough endoplasmic reticulum. In the absence of the ribosomes, they appear smooth and are called a smooth endoplasmic reticulum. RER is frequently absorbed in the cells actively involved in protein synthesis and secretion. They are extension and continues with the outer membrane of the nucleus. The smooth endoplasmic reticulum in the major sites for synthesis of lipid. In animal cells, lipids like steroidal hormones are synthesized in AR.